I am R. Parvati, St. Mary's College, Department of Microbiology, 2nd BSc, Molecular Biology and Microbial Genetics. Assignment topic LAMDA, PHAGE and their life cycle and structure. Application of bacteriophage.
Welcome to Molecular Biology and Microbial Genetics. Macrophages are the type of the white blood cells circle for the immune systems, acting as savages that engulf and destroy pathogens and cellular describes through pathogenetics. Bacteriophage virus that inject bacteria can be excreted to main life cycles. Lytic and the lysogenic lytic injection bacteria phages rapidly replicated within the host cells, eventually lyses it to the release new phages particles. In contrast, the lysogenic phages integrate their DNA into the lysosome host's genome, become a bacteriophage. They infect equally either the lytic or the lysogenic cycles may be followed into the cases of the first step involves in the phages attaching to the host cells and then infecting in the DNA into the host cell in the lytic cycle. Bases of nucleic acid in the replications and the phagogenics are expressed in the resulting of the productions of the phage proteins matured phages of particles on the assembled and the host cells lysis after releasing the phages of the particles. In the lysogenics of the cycle, the phage DNA is not replicated or the transcriptions. Instead, the phage be in the lysogenic state when the cells are exposed to the ultraviolet light. The certain of the chemicals bases the proposed DNA excreted and phage enters the lytic cycle. Virus is the passage cell. It attacks the host cell and takes over in the chemicals actually for the making the proteins and the nucleic acids. One of the better understood virus life cycles in the phages, a virus which attacks the bacterial cells. The phages begins its assault but after attaching itself the cell wall of the bacteria. This attachment is very specific and each type of the phage can only be attached to the certain type of the bacteria. Next, the phage beats down the cell wall to form the capsule injected through the hollow tail into the cell. The viral nucleic acid uses the cell enzymes and the organelles to the replicated new viral pods. The pods are assembled into the new virus particles cells viruses. Finally, the new phages of the releases and the enzyme and the host cell hosts releasing the virions. Up to 300 new virus particles can be reproduced in a single host cell. This process of the viral multiplications in the called the lytic cell. Interestingly, the lytic cell does not always happen immediately. Sometimes, rather than the producing virus phages, nucleic acid incorporates in the host cell DNA. Cell to divides the phages DNA as also reproduces and is spread to more and more cells. Then suddenly for reasons that are still unclear, the phage's DNA activates and the destructive lytic cycles begins. Once again the host cells destroyed and a new generation of the phage are released invaded new hosts. Phage are being exposed for the various applications. Phage therapy using phages treatment bacterial infections, especially those instant to antibiotics. Biotechnology utilizing phages for the virus research and the industrial purpose. Diagnostic tools developing phage-based assays for detecting and identifying bacteria. Notes for lambda phage and their life cycle structure applications of bacteriophage. Most abundant biological particles in water and the second most abundant component of the biomass on the land following the prokaryotics. The phages of the sizes of the phages range between the 24 to 200 nanometer in length. The shape, size and structure of the different bacteriophages are the different. Had a short collar and the helical tail. Phage anatomy. Head, tail, ring, facial plate, corner. Life cycle of bacteriophage. Lytic cycle or virulent cycle. Lysogenic cycle or temperate cycle. Applications of bacteriophage. Treatment of bacterial infections. Food hygienic and safety. In agriculture.